Our next speaker is Thom Saunders from Michigan, CRISPR Cas9 genome editing pipeline for mice and rats.
I want to thank the organizers for inviting me to present our work. This is the outline of a CRISPR Cas9 project. We begin by designing the sgRNA to the target, clone it from oligonucleotides. Then we go on to test it in cell culture. We'll also test it in zygotes to make sure it's active before we go on to make an animal model. Then it's just a question of genotyping, germline transmission, and analyzing the phenotype. So Prem gave you the introduction to CRISPR Cas9, so I'm not going to spend time on that. The main thing we're using Cas9 for, what we want it to do, is to produce a double-strand break on the chromosome. And once we have a double-strand break, whether it's induced by CRISPR, zinc fingers, talons, or meganucleases, the chromosome in the zygote has to undergo either non-homologous end-joining repair, in which case an indel of a few base pairs to a few hundred base pairs can be formed. This typically will knock out gene function if you chose the correct location in the genome by nonsense-mediated decay of the messenger RNA. Alternatively, if you introduce a DNA donor, whether an oligonucleotide donor or a double-stranded DNA donor, you can now introduce new information into the genome, such as a point mutation, a cDNA, a fluorescent protein, a recombinase, CRE, FLIP, DRE, etc. So this is where we go to design the guide RNAs. This is a website that amalgamates data from 8 other websites using various algorithms. This was published this month, so if you're interested in more details about this particular algorithm, I invite you to refer to this publication. It's really handy. One feature it has, it'll tell you if there are any other potential off-targets on the same chromosome you're working on, and that makes it easier to screen for off-targets in the downstream process after you get germline transmission. So it's pretty simple. You design a couple of complementary oligonucleotides, anneal them together, drop them into the BBS1 enzyme sites in the PX330 vector that Feng Zhang developed. We had a lab on campus who actually had a high school student doing this work, but high school students tend to go back to high school in the fall. She went back to high school, and then we had some eminent postdoctoral research fellows that picked up the project, and after we got their reagents, like we would do the microinjections, no mice would be born, and so then we realized we have a problem, and so message to the postdocs in the audience, just follow the protocol, don't try to improve it. Okay. So once we went back to the original protocol, then we got good results, and we got viable embryos. So that is one of the reasons we do the zygote microinjection, because if the lab drops off the CRISPRs, we microinject it, there's no pups born. We want to know that the reason is not toxicity at the zygote stage. So in terms of screening in cell culture, it's pretty simple. You take some primers that go across a predicted cut site, PCR amplified DNA from the transfected cells, subject it to a T7 1-endonuclease assay. It's a gel-based assay. You rapidly get an answer. So in this example, this is DNA from wild-type cells that were not treated with CRISPR. And in these cells, if we add T7 endonuclease, you'll notice there are no extra bands appearing. And even though we're using, C57 black 6N cells, sometimes we'll see extra bands in the wild type with this assay, indicating there may be some spontaneous point mutations popping up, or the primers are amplifying something weird, so you may have to optimize the primers. If we introduce a guide and CRISPR Cas9, then we'll see new bands appearing of predicted sizes, because we know where the cut is expected to be. We know what size bands to expect. This tells us we have activity. In cell culture, when we test in zygotes, we'll culture the zygotes to the blastocyst stage, again amplify across the cut site, and then subject the PCR product to DNA sequencing. This is a typical result a sample where we see a nice, clean DNA chromatogram with no background, and then we get to the predicted cut site, and now you can see we've got peaks on peaks in the sample, indicating there are multiple DNA templates, and these are being produced by the indels. And the sequencing technician says, sorry for your lousy run, you gave us a sample that was contaminated with multiple templates, but that's okay, that's what we want to hear. And so then some basic principles on designing the donor. The DNA donor, whether it be an oligonucleotide or a double-stranded plasmid, should block the Cas9 cleavage. You don't want cleavage after this zygote picks up the donor. Small changes such as point mutations, LOXP sites, epitope tags can be introduced with oligonucleotides, 150 to 200 base pairs. You want to make sure that you get the ultramers that have reduced chance for errors in the oligos, so you don't get out of mouse and then discover, oops, there's a missing base pair in my LOXP site. I wonder why this gene isn't conditional. We generally add restriction enzyme sites to make screening easier. We can have dropped in CRE, FLIP, GFP, fluorescent proteins with circular double-stranded donors with 5' and 3' arms of homology, similar to gene targeting in yes cells, although in zygotes, of course, there is no drug selection. The arms are short, so we basically use long-range PCR screening, because when I talk to the postdocs and I say, how about a southern blot screen, all the postdocs get up and leave the meeting. So it's all PCR screening. So here's some examples of knockouts. And so we injected a bunch of eggs, we got out a bunch of pups. So the key thing you want to focus on is this column on the efficiency. So if we just define the efficiency of the getting a gene-edited mouse as the number of gene-edited founders per 100 micro-injected zygotes, we can make comparisons across genes, across Cas9 reagents, even across species, and across experiments in the published literature. So this is a handy metric that you can use to analyze data in a paper or in your own lab. And so you can see in just producing indels with a single Cas9, the efficiency range between 1.2 to 9.5 founders, which is sufficient for knocking out most genes, all genes, as long as you put the indel in the right place. And then for those people who require inbred mouse strains, i.e. C57 Black 6J, or a mouse strain from your own personal colony, or a sensitized genetic background that always is already carrying multiple mutations, it's probably going to work, but with less efficiency, right? So if we use a hybrid background in this experiment, we got out 30 founders per 100 micro-injected eggs. So as an embarrassment of riches, we take the identical CRISPR reagent, we drop it into black 6 zygotes, you'll notice the efficiency drops. So yes, it is possible, it's just going to be less efficient. We've also used CRISPRs to take out regulatory regions for some experiments, and the efficiency of taking out a DNA segment with 2 guides is equivalent to taking out an exon with a single indel. So 1 guide, 2 guides, similar efficiency. And here are the knock-ins. So the oligonucleotide knock-ins, this is one example. We've done a number of these. The efficiency is basically the same as for a standard indel. But once you go to put in a knock-in, then it's less efficient. So in this M-Cherry example, we got out 1 pup for 303 micro-injected eggs, and this DNA donor with multiple amino acid variants, it was 1 pup out of 306 zygotes. In this example of CRE, it was 1 pup out of 297, and you'll notice this one was not successful. So there are a number of reasons for this, such as mostly locus-specific effects, the design of the gene targeting vector, and then the sensitivity of the screen, because typically with a PCR screen with an inside primer and an outside primer, you don't get a chance to validate the sensitivity of the screen before you get to the mouse stage. These are examples from the rats. So if we use CRISPR to make indels in rats, the efficiency is the same as the mouse, between 2 and 10.7 gene-edited rats per 100 microinjected zygotes. Oligonucleotides, again, are efficient. Here's an example of a CRI. It's actually more efficient in the mouse. We've also done another CRI and another flip Knockin in rats. The efficiencies seem to be slightly better than in the mice for reasons I can't explain. It may again be allele specific. So here are some examples. This is an example of a phenotype in a mouse model where we used an oligo-Knockin to produce a truncated protein to model the situation observed in human patients. So in these mice, we see retinal dystrophy, right? The outer segment is compacted compared to the wild type. And so patients, this may be one of the reasons for their pathology. The lab is now continuing to pursue this. And then this is the example of the cherry Knockin. We designed a couple of guides. We took the most highly active guide and used a targeting vector with 1.7 kb 5' arm, short arm of only 331 base pairs. And this is the one Knockin mouse we got. And the lab was happy because now the cell population they're interested in studying is expressing M. cherry, as can be seen in this flow cytometry data. So now they're going on to do their studies in their mouse. This is an example of a floxed allele. It's the same strategy. We make a single cut on the chromosome. We add a targeting vector. This one had an 800 base pair 5' arm, 338 base pair 3' arm. We had a diagnostic restriction site, and we obtained one animal that was identified as being floxed by the presence of the Bagel2 site. The lab went on to confirm the presence of the second LOXP site in cysts, and now they're breeding up these animals to their favorite Cre recombinase mouse to produce their model. And so in terms of future directions, the field is moving rapidly. This year, we had the enhanced specificity Cas9 published. We had the high fidelity Cas9 published. And then we have the some long Latin name, Argonaut DNA guided nuclease. And I was talking to Gaetan Borgio, who has a talk later today, showed me some interesting preliminary data with Argonaut. So next year, we may be giving the same talk with a different nuclease because there are some advantages in being able to target GC-rich templates, and we don't have the limitation of a prototypic adjacent motif telling us where we can cut. In terms of the field generally recognizes that homology-directed repair is the bottleneck. People have tried the SCR7 small inhibitor of DNA ligase 4 to knock down non-homologous end joining, and our experience is, yes, it knocks down non-homologous end joining, but it doesn't do anything to increase homology-directed repair with the DNA donor. So recent data is suggesting that RS1, a stimulator of RAD51, will actually stimulate homology-directed repair, and that's where a lot of labs now are pushing in the field. They're pushing on RS1, they're pushing on the Argonaut system. And these are my colleagues in the lab, the people who are doing the microinjection, building the reagents, doing the genotyping, managing the animal colonies, etc. Thank you for your attention.
Okay, questions?
So for delivery methods, can you say differences in success between electroporation and microinjection?
The principal difference between electroporation and microinjection is that electroporation is more gentle, more zygotes survive, more pups are born. Yeah.
I'll just stand up and yell. So what's the minimum arm length that you see that's necessary? Have you done a test of arm length versus...
We have not done a systematic survey, but that would be a good idea if somebody wants to provide me some info. Yeah.
Question was
Well,
what
the question was the minimal arm lengths of homology. So I gave you a couple of examples where the arm lengths were 300 base pairs on one side, 800. So that's probably the shortest we've gone. It's like 300 and 400 base pair arms.
So, question, Thom. The limitations of or the inefficiency of black 6 is a problem given people's expectation that you make a black 6 mouse, God forbid you don't. Is your recommendation just to keep trying, or do you make the mutation in ES cells first and use a proven ES line?
Yeah, so we can do, so if it's not working in black 6, then we can go to ES cells with a gene targeting vector. So we designed these donors so they have a convenient site where we can drop in a drug selectable marker and go into ES cells with the same CRISPR to stimulate homology-directed repair in the ES cells. And then we have the advantage of drug selection, so we can screen many ES cell clones, or we can have a lot more ES cell clones in an electroporation cuvette, right? Millions compared to a few hundred zygotes. So it gives us, yeah. So then we'll take those clones, pick a plate of clones, find the positives, and go on to mice. But to be honest, the scientific community is kind of running away from ES cells. The demand isn't there.
Okay, very good. Thank you very much.